In this module, we're going to describe how reads are generated under this model where we have different transcripts for the same gene. And then we'll use this to explain the statistical models that are used to quantify them in a next module. So to describe the type of data we would see in a typical RNA-seq experiment, we're going to use a cartoon where we represent one gene that has 3 exons, and in this case has 3 transcripts represented in the sample. So we have transcript 1 is composed of exon 1 and 2, transcript 2 is composed of all 3 exons, and transcript 3 is composed of exons 2 and 3. These transcripts are present at some level. In this cartoon, we're acting as if the first one is at 2, the second one is at 1, the third one is at 2. So these transcripts are present, they get turned into cDNA, and now we have reads after we sequence the cDNA. So here are the reads. So notice we have some purple reads because they come from exon 2, some red reads that come from exon 1, some yellow reads that come from exon 3, and we also have reads that are coming from the junctions. These are the ones that are multicolor. So we have the red and purple coming from the junction between 1 and 2, and the purple-yellow coming from the junction of exons 2 and 3. So these reads here on the left, we're thinking come from this transcript because we have red, purple, and junction reads. These reads are coming from this transcript, and we have yellow, we have red, purple, we have purple, yellow. We could have a red as well, although we didn't see one here. And then the same thing goes for the third transcript. Now, the question that we ask now is, can we reconstruct these transcripts? Because we don't know a priori that these were the transcripts that were there. All we see are the reads. So this is the challenge. And then a second challenge is, can you then quantify how much of each transcript is present? That's difficult. So there's different approaches to transcript building. There are approaches that assemble the transcript without a genome reference. Examples are Trinity, Velvet, and OASIS. These start with the FASTQ files. You just have the reads and you try to assemble the transcripts. A second approach is to use the genome, and that's the one we're going to explain here. Cufflinks and scripture are examples of this second approach. So the idea is relatively simple to explain, although hard to implement. You have the location of exons. So what you do is you map the reads to the genome, taking into account that some of these reads might be split between 2 distant parts of the genome. For example, in this cartoon, these reads here are such reads. Those are the junction reads. So when we map them, we have to be mindful of that this part of the read is going to go on this part of the genome, then there's a gap, and then this part of the read is going to go on this part of the genome. So we're mindful of that and actually map reads this way. So when we're done with this approach, we know which read comes from which exon and which reads come from which junctions. So example of software that does that is TopHat, GSnap, MapSplice, Star, PalMapper, and there's others. There's many others. So in the next module, we're going to explain now, once we have this, how do we quantify how much of each transcript we have. But before doing that, I just want to explain that once you're done with these mappers, you can also use this information to create the gene count tables. Right? So you can basically count all the reads that fall into a gene, regardless of what transcript they come from. They come from the exons from this gene. And you could also do counts at the exon level. This is a relatively simple exercise, and we showed you in one of the modules how we do this. This creates the table where you can then do things like differential expression. You have a table of counts for either exons or genes on the rows and samples on the columns. On the next module, we're going to start explaining the more complicated step of quantifying transcript levels.